All right, so we are going to be talking about active transport. This is Chapter 3, Section 5. Follow along in your textbook and take your Cornell notes in your interactive notebook. Active transport is going to be the basically opposite of passive transport. So it is transporting membranes against the concentration gradient. So we're going from low to high. So I would suggest putting a diagram like this in your notes. Here you again are going to have your membrane separating the two sides. Here's your low concentration. Here's your high concentration. And now we're trying to jam more molecules into a place where we already have a high concentration. So because of this, active transport is going to require energy in the form of ATP. ATP actually stands for adenosine triphosphate, and we're going to be talking more about that next unit. So what pumps these materials against the gradient again are called protein pumps, okay? All transport proteins are going to change shape, and again, they need energy in the form of ATP. So we're going to look at the sodium-potassium pump as our example of an active transport pump. Here's an example, a simplified example of a transport protein pump. Again, here is our low, here is our high. We need to have that energy input in the form of ATP, shown kind of like this or in a lightning bolt. Energy is needed by the cell to make this transport happen. All right, we also have examples of what are called bulk transport. Bulk, if you buy in bulk, oops, why is that doing that? If you buy in bulk, you're buying things in large quantities. Same thing with transporting big items. It's not just going to go through these pumps or through the phospholipid bilayer. We're actually going to transport them in vesicles, okay? So this is movement of substances into the cell by a vesicle. This also requires energy, okay? And there's two types, moving things in or moving things out. Endocytosis is going to be moving things into the cell. So what's going to happen is that the substances are going to approach the surface of the cell membrane, and remember it's a plasma membrane, so if this is inside the cell, this is outside the cell. Here are our big molecules, a bunch of them that we want to get into the cell. What's going to happen is that this is actually going to come up and kind of form a little vesicle, pinch it off, and then bring it into the cell. Here is a better diagram of that. So here's our stuff we want to take in. Plasma membrane's going to come, it's going to kind of pinch it off and then bring it in a vesicle. Remember, the vesicles are things that are transporting things around the cell. They're like the little vehicles. Right, very similar thing happens with exocytosis. Now we're taking things, this is out of the cell, exiting. Okay, I always think of exiting the cell. The vesicle, again, is carrying the contents which we want to spit out of the cell. Again, these are bigger things that would be able to, we're not going to transport them through the phospholipid bilayer. We're not going to put them through transport proteins. They're going to come up to the plasma membrane. This is going to fuse. It's going to open up, and the contents or whatever's inside that vesicle is going to be spit out to the outside of the cell. That is exocytosis. So the actual definition of this is the release of substances out of the cell by the fusion of a vesicle with the membrane. So exo is meaning out. All right. We'll talk about later on this year nerve impulse transmissions. We have these things called neurotransmitters that get to the end of one neuron and then need to be passed to another neuron. So that's a specific example of exocytosis. Right, and that does it for the cell function screencast. This was number 3 of number 3. And I'm doing this one at school, so there was no chance of Roxy making an appearance. Sorry. We'll see you in class.